All right, so good morning, everyone. I would like to thank the organizers for giving me the opportunity to present my work here. So first of all, I would like to start with a brief introduction to the immune system. So essentially, the immune system can be divided into two major parts. You have the innate immune system and the adaptive immune system, where the innate immune system is responsible for a first quick line of defense against invading pathogens, and the adaptive immunity, which is slower but more specialized, will try to catch any pathogens that manage to slip through the innate defense. My talk will be focusing on the cellular part of the adaptive immunity, so I'd like to briefly expand on that as well. So imagine you have one of your cells being infected by an intracellular pathogen. Your cell will, as a response, try to process this pathogen into small bits or peptides, which will then be presented on the cell surface through a class of molecules called major histocompatibility complexes, or MHCs. And this MHC-peptide complex will then be surveyed by circulating T-cells from the immune system, and whenever an MHC-peptide is recognized that is presenting a non-self peptide, this will activate these T-cells and cause an immune reaction. So the question that we ask ourselves is it possible to actually predict whether a certain T-cell receptor will recognize such an MHC peptide, yes or no? However, this is not a very simple problem. There are some difficulties with this. First of all, because it has to recognize such a large diversity of possible pathogenic epitopes, there is also large diversity in the immune system itself. So for example, the presenting MHC molecules, there are about 8,000 almost, I think, known classes of MHC molecules, class 1. The peptide itself can, of course, be very diverse. Usually presented peptides are about 9 amino acids long, so you can calculate how many possibilities we have there as well. And to make it even worse, the T-cell receptor itself consists of 2 chains, an alpha and a beta chain, where each is created through a process of semi-random recombination. And for example, beta chain diversity is estimated to be able to generate about 10 to the 12 to up to 10 to the 15 possible different chains. It's slightly lower for the alpha chain, but you can imagine that combination of both results in a tremendous diversity as well. So this is a bit of the theoretical challenges, but we still went and looked in the literature for some data as well. This presented some challenges of its own, since there isn't a lot of TCR epitope interaction data known, but we managed to find some nonetheless. Some more challenges that we faced is that, as I told you, the TCR consists of both an alpha and a beta chain, where we have here these semi-recombinated parts that will do the epitope interaction within the CD8 region. But usually, when sequenced, we only have the beta chain, and it's further complicated because the CD8 region, due to the semi-random recombination, can also have a variable length as well. So how did we manage these challenges? We tried to create some sequence-based features. We incorporate, for example, which V or G gene segments were used, as well as the length of the CD8 region and the amino acid composition. In addition to this, we also calculate some averaged physicochemical properties over the CD8 sequence. In addition, we also create a lot of position-specific physicochemical properties, where we started from the center of the CD8 region and then went towards the end. So how did we tackle this problem? At first, we tried to distinguish whether a population, we could divide a population of T cells with a TCR receptor could be divided into recognizing either of 2 peptides, like this. And surprisingly, due to difficulty, even with the difficulty of this problem, we managed to still get a pretty decent performance out of this. So we challenged ourselves a little bit further, made the problem a little bit harder. This time we started from a more diverse repertoire, where T cells could either bind this specific epitope or another epitope that wasn't known. And we repeated this for both epitopes that we had. And even there, we still could get a pretty decent performance for each of the classifiers. Because we used random forest classifiers, we were also able to extract some of the important features that played a role. And we see that the majority of features that seem to determine whether a T cell will bind a certain peptide or not are based on the physicochemical properties. And to give you a little bit more insight in this, this was mostly found to be either near the center of the CDR3 region or averaged over the whole CDR3 region. We also tried to look whether we could increase the performance even further if we would have more data available. So to do this, we subdivided our training samples into smaller parts and then slightly increased this and checked the performance each time. And as you can see, for all the 3 classifiers that we tried, the performance still slowly goes up and hasn't reached the plateau yet, which indicates that some performance could still probably be gained if we have more available data. Some of the take-home messages that I would like to focus on is that we provide a proof of concept for predicting interaction between T-cell receptors and an epitope, even just only using the beta chain and based on primary sequence features. So we definitely show that this is a possible problem. It's feasible to solve this. We also show that with the increase in data, some performance can still be gained. And this research opens up, or knowing that this is possible, opens up a lot of opportunities in diverse immune-related domains, for example, such as in immuno-oncology. With this, I would like to conclude by thanking my supervisors, Chris Laukes and Peter Meisman, as well as the other members of our group. If you're interested, we have a poster this afternoon, which you can find here, or you can read the bioRxiv paper here as well. Thank you for your attention.
That was an interesting talk, thank you. I was just wondering if you have considered to remove the information about the V and J genes from your model and if you can maintain performance, because many sequencing projects just target the CDR3 beta sequence.
Yeah, definitely.
And I was also wondering if you have performed any homology reduction or clustering on your data set prior to partitioning it into training and testing.
So for your first question, we did look a bit into the V and the G genes, and we see that within the feature importance of our model, these are actually, they have very, very low importances, and it's mostly the physicochemical properties that seem to determine whether one of the 2 peptides is actually bound. The reason that I suspect that this is less important in this case is because we kept the MHC background constant, so we always have B08 as a MHC background, and this might be more important when you're looking across different MHC ranges. Could you repeat the second question? Sorry.
Yes, sure. So I was wondering about homology reduction or clustering of your...
We haven't looked at the homology itself between the sequences, but during cross-validation we made sure to randomly sample a lot of times. So I think it's highly improbable that maybe some similar sequences cluster together in the training data versus testing, for example.
Can you just tell us how many, what the size of the data set is, how many TCR sequences, how many epitopes?
So for the problem where we tried to distinguish the 2 epitopes from each other, we had about 100 TCR sequences for each epitope. And when in the case that we tried to distinguish an epitope from our background population, we had a lot more background TCRs in a few thousand, as to control a bit for.
Follow-up question. I understand you did a lot of work to extract this data. Did you now organize it in some more sensible fashion? Did you put together some sort of database?
So the data that we used is available actually. We also have a GitHub page where you can find all the code and data that we used. You can also find it in the bioRxiv paper as well. So if you're interested, definitely have a look at there.
Thank you very much.
You're welcome.